Hello everybody, my name is Taipe Huisa, and today I will present you my e-poster entitled Cytotoxicity of acai silver nanoparticles for melanoma and leukemia cells with and without multi-drug resistant phenotype. So, as introduction, it's important to know that acai is a plant that possesses a high content of polyphenols and flavonoids, which make them very suitable for the synthesis of silver nanoparticles through a green synthesis process. And on the other hand, in the last years, there have been many studies that have explored the potential use of these green synthesized nanoparticles on cancer treatment. So this was the reason that the aim of this work was to evaluate the cytotoxicity potential of silver nanoparticles, in this case synthesized with acai, against melanoma, B16FT stem cells, and infiltrate leukemia cells, K562 and FEP cells. And this second one, the FEP cell lines, have a specific characteristic, which is that they possess an MDR phenotype. And this MDR phenotype is because they possess a higher amount of a protein responsible of the extrusion of chemotherapies, which is the glycoprotein P or PGP. So, how do we do this? First, we synthesize our silver nanoparticles with acai using a thermal process already standardized and published. And after this, we perform all the cytotoxicity assays using the MTT viability assays on all the cell lines I mentioned before. And then we perform molecular talking analysis. And this we did this to check if there was any interaction between the protein responsible of this MDR phenotype and our silver nanoparticles. And finally, we perform a PGP activity experiment to see if actually we could modulate the activity of this protein with our treatment. And for this, we use the rhodamine extrusion assay. So, in terms of the results here, first you can observe in figure 1 our nanoparticles produced with acai, which have approximately 20 nanometers in size, as obtained by electron microscopy. And then the MTT assays have shown that all the cell lines tested are sensible to our silver nanoparticles, which are actually very cytotoxic against all of them, and specifically the leukemic cell lines. And something that was very interesting to observe was that the FEP cells which possess this MDR phenotype are also sensible to our silver nanoparticles. That's why we perform the docking analysis to see if the inhibition of the PGP protein would be a possible mechanism of action. So here we perform docking analysis and we observe a strong interaction actually between this protein and two compounds in the acai, which are cyanidine-3 glucoside and cyanidine-3 rutenside. And finally, we perform the actual experiment with the rhodamine extrusion assay, and we observe that the exposure of silver nanoparticles in the FEPs actually inhibit the activity of the PGP. So the conclusions of this work overall show that our nanoparticles have a high cytotoxicity against melanoma and leukemia cells, including the MDR variants, talking about the leukemic cells, the FEP cell lines actually. And this cytotoxicity might be through the modulation of the PGP function. And these results strongly suggest that silver nanoparticles synthesized with acai might represent a promising alternative for treatment of both MDR and non-MDR cancers, but obviously more research has to be performed. Finally, I would like to thank to my university, the Federal University of Rio Grande, and the post-graduation program of physiological sciences where this work was performed, and also to the finance agencies, which are CAPES and CNPK. Thank you.